Okay, so welcome to this next video in the playlist on cyclic AMP signaling. And in this video what we're going to do is we are going to look at the A Kinase Anchoring Proteins. So the topic for this video is A Kinase Anchoring Proteins. And basically what these A Kinase Anchoring Proteins give rise to is two types of protein kinase A, namely protein kinase A1 and protein kinase A2. So protein kinase A1 is basically protein kinase that is free within the cytoplasm of cells, whereas protein kinase A2 is not free. It's anchored somewhere by an A kinase anchoring protein. So the format I'm going to take for this video is, firstly, we're going to look at protein kinase A1 in a bit more detail. We're going to see its structure, and we'll have a bit of a revision of the previous video on introduction to protein kinase A, and we'll see how it's free in the cytoplasm and how that means that when it's activated by an increase in cyclic AMP, it's free to activate a lot of different effectors, basically. Then what we'll do is we'll look at A kinase anchoring proteins and how they give rise to the second type of protein kinase A, namely protein kinase A2. So let's start off with protein kinase A1 then. So protein kinase A1. And I should also say that there is another name for protein kinase A, which is it's also often called the cyclic AMP dependent protein kinase. So if you ever hear someone talk about the cyclic AMP dependent protein kinase, what they are talking about is protein kinase A. So cyclic AMP dependent, oh dear, I missed a word out, protein kinase. And this would be the cyclic AMP dependent protein kinase 1 then. Right, okay, so I'll just put a little equal sign there. So this is protein kinase which is free within the cytoplasm of cells, so it's not bound anywhere. So if we have a cell here, basically protein kinase A1 is just free and suspended in the cytoplasm of cells. So here's a cell, and inside it obviously has a lot of cytoplasm, and in the cytoplasm will be protein kinase A1 subunits. And basically, their structure consists of this regulatory subunits, this dimer of regulatory subunits with catalytic subunits, so the R2C2 complex. Okay, so we discussed the R2C2 complex in the previous video. In this video, I'm going to draw a slightly different cartoon for it, but it basically is the same principle. So I'm going to draw two regulatory subunits, but I'm going to draw them in this nice little shape here, sort of bent over. And here are the two cyclic AMP binding domains on this single regulatory subunit. So this is one of the regulatory subunits. Now it's a dimer of regulatory subunits, R2. You have two regulatory subunits in. So here's the second regulatory subunit with its two cyclic AMP binding domains there. Okay, so the two regulatory subunits have dimerized together, and now basically the catalytic subunit is held to the regulatory subunit. So this is a catalytic subunit, this is a catalytic subunit. So this is the R2C2 complex because you have two regulatory subunits with two catalytic subunits. So this is a protein kinase A1 molecule. So protein kinase A1. Okay, and the reason it's protein kinase A1 is because, well, firstly, it's not suspended anywhere, and the reason it's not suspended anywhere is that these regulatory subunits are a specific type. They are the regulatory 1 subunit. So you basically have two regulatory 1 subunits in protein kinase A1, and these regulatory 1 subunits don't have the necessary binding domain to bind to an A kinase anchoring protein, which would anchor them somewhere and fix their position. Okay, so when cyclic AMP goes up in the cell, and I will draw cyclic AMP as a little blue ball again, when cyclic AMP goes up in the cell, what happens is that cyclic AMP is now going to bind to these 4 cyclic AMP binding domains. So what happens basically is, often it's drawn in cartoons like so. The cyclic AMP binds, and that changes the conformation of the regulatory subunit. So I'll show That's here, and that's why I prefer this cartoon actually to the one that I drew in the previous video, because this shows you a nice conformational change that has actually happened. Basically, they're straightening out. Okay, so here are our two cyclic AMP binding domains, and now they're occupied by cyclic AMP. Right, so these are still regulatory 1 subunits, which means that they don't have the necessary binding domain to bind to an A kinase anchoring protein. And here is 4 cyclic AMP molecules bound in those cyclic AMP binding domains. And now, basically, in this state, they no longer sequester the catalytic subunits. The catalytic subunits go off. Okay, so you've now got 3 catalytic subunits just floating around in the cytoplasm. Now, when the catalytic subunits were bound to the regulatory subunits, they were inactive. So these weren't catalyzing any phosphorylation reactions at all when they were bound to the inactive regulatory subunit. When the regulatory subunits bind to cyclic AMP, they dissociate from the catalytic subunits. So the 2 catalytic subunits are now free in the cytoplasm, and they are now active, basically. So these are active catalytic subunits. Okay, and basically the reaction which they catalyze is they catalyze the phosphorylation of serine and threonine residues. Okay, so they will go to loads of different effectors in the cytoplasm and in the cell membrane, and they will add phosphate groups onto the serine or threonine residues of the amino acids in those they'll add phosphate groups to serine and threonine residues in the polypeptides, basically, and that can change the function of those polypeptides. So these are going to act as serine-threonine kinases, and we'll just have a little bit of a revision of the structure of serine and threonine, which are the amino acids that these catalytic subunits are going to add phosphate groups onto. Okay, so let's just go down here and do this bit of revision. So the basic amino acid structure is that you have an amino terminus up here, okay, then you have the alpha carbon here with a hydrogen, and then the carboxyl group down here, like so. Right, and then in serine, the R group is a methylene group with a hydroxyl group on there. So that's serine. Now, in the case of threonine, you have the same core amino acid structure with the alpha carbon and the amino and carboxyl terminal domain. Amino and carboxyl domains. And then you have, off on the R group, you have, it's a very similar group to serine. You have a single carbon with a hydroxyl off it, but instead of one of these hydrogens, you have another methyl group sticking off there. So that's threonine. So basically what these catalytic subunits do when they are active is they phosphorylate these hydroxyl groups. So what they'll do is they'll take off this proton and bind instead a phosphate group on, which is a group that looks like this. Okay, so that's a phosphate group, and you can see that this is a very interesting group because it has this negative charge here. It has in fact a lot of polar residues here, polar groups. So these are all quite reactive, so this may well change the structure of the protein now that you've added this phosphate group. And that can change the activity. Now, the important thing to understand in the case of protein kinase A1 is that these active catalytic subunits are now free in the cytoplasm. So they will go and do this to a great host of effectors, basically. So they will affect the function of a huge number of proteins. And we're going to contrast that to protein kinase A2, which is made out of a different regulatory subunit, namely the R2 regulatory subunit of protein kinase A, and that regulatory 2 subunit of protein kinase A does have a binding site for A kinase anchoring proteins and will be anchored in a specific position, and basically it will phosphorylate only the proteins that are in its vicinity, basically very close by it, because it's anchored in position. Okay, so in the next video we'll have a look at protein kinase A2.